Hello everyone, I will be speaking on the topic bacteriorhodopsin. So bacteriorhodopsin are present in photosynthetic archaea and they have been researched extensively. Therefore, the function shape is now well known and shape of the membrane protein bacteria. Rhodopsin has both alpha helix and beta helix. Alpha helix can be seen, you can see in the green, and the beta helix it is seen in the red. The alpha helix have hydrogen bond binding the amino group to the backbone and have the side chain facing outward. The beta strand are platelet and also have hydrogen bond bonding the amino acid the chain to the backbone, but beta strands have the side chain. Alternative, it has inward and outward and can be parallel or antivalent in the structure. We can see the secondary structure of alpha helix and beta strand combines to forms a tertiary structure of the bacteriorhodopsin. To continue this, there is a membrane protein, so it has to interact with the this one has to interact with the phospholipid bilayer. Phospholipid bilayer, it is amphipathic, which means it has both hydrophobic and hydrophilic part. In this membrane, we can see that hydrophobic parts and the hydrophilic part. The hydrophilic head and the hydrophobic tails are very important to this phospholipid bilayer stuff. And this protein, the bacteriorhodopsin, has to be very flexible in order to interact with both hydrophobic and hydrophilic parts. This membrane protein have very similar function with the sodium channel pump in the human nervous system. Bacteriorhodopsin pumps out ions that are inside. So you can see here bacteriorhodopsin pump out the ions that are inside the cells to the outside. This is the outside part and that's the inside of the cell. The cells to create a. So what does bacteriorhodopsin do? The bacteriorhodopsin pumps out ions that are inside the cells to the outside of the cell to create a potential gradient between the inside and outside of the cell. This bacteriorhodopsin is able to function because of the conformational change that occurs when sunlight hits the bacteriorhodopsin. Right then there is a change its shape and then it makes the bacteriorhodopsin able to pump the proton outside of the cell. Conformational changes is essential. Conformational changes is essential to function as protein will have their shapes to be effective. This is the same here where there is no sunlight. The proton will not change the shape and therefore will not function correctly. So it is important to have sunlight.